All right, welcome everyone. We'll go ahead and get started. Thank you for coming to today's EEB seminar. We're going to get started. I think Nandita probably has like 400 slides to get through, so we should get started. It is my distinct pleasure to introduce Dr. Nandita Garud, who's, I assume, familiar to all of you. She's today's seminar speaker. So Nandita has been with us in EEB since 2019. And as when she started her faculty position here, as you may know, I first knew Nandita when she was an undergraduate at Cornell, and I was a graduate student at Cornell. We were both in the Clark lab, and it's been great to watch her career develop and take off since those days. So after her undergrad at Cornell, Nandita went to Stanford, where she did her PhD with Dmitry Petrov, and really shook some paradigms in population genetics about the importance of soft selective sweeps in evolution. But rather than just wanting to continue on with Drosophila genetics for her postdoc, she went to work with Katie Pollard at UCSF and the Gladstone Institutes, where she took on a whole new sort of category of data and questions in terms of evolution in the microbiome. And so she came to UCLA, as I mentioned, in 2019, where she's sort of combined these interests of population genetics and selective sweeps and adaptation with evolutionary and ecological processes in the microbiome. And so that's how what she's worked on in her research program since arriving here. She has won a number of distinct awards, probably most famous and impressive of an NSF Career Award. And yeah, Nandita's been a great colleague and teaching a number of quantitative courses in our department. And yeah, really excited to hear her tell us about her lab's research. So with that, Nandita.
All right, well, thanks for coming, everybody. I really appreciate the show of support. It's my great pleasure to share research from my group today on the ecological and evolutionary dynamics of the human gut microbiome within and across hosts. So before I begin, I'd like to first thank the members of my lab, some of whom are not pictured here, actually. They're former members too. And they've created a really exciting intellectually vibrant environment in my group and make coming to work really a joy. And so I'm going to be introducing their work as we go along, one by one. But before I begin telling you about the research specifically going on in my group, I want to share with you the broader research agenda that really motivates me and my team. And that is, how does adaptation proceed in natural populations? So we've been really interested in natural populations because you cannot quite capture all the vagaries of nature in a test tube or in a simulation. There could be fluctuating temperatures, fluctuating diets within a host, and these are all forces that are affecting organisms on this planet on a daily timescale, monthly, minute, all the time. And so it's important to be able to quantify what's happening outside in nature, but then also to make sense of it in the context of what's going on inside of the lab. And so my group studies a whole variety of organisms. In my past, I've looked a lot at Drosophila, and Mariana Harris in my group continues to do so. We also are looking at ancient humans and recently worked on looking at selection in New York City rats. But a massive focus in my group is looking at evolution in the human gut microbiome. Why the human gut microbiome? Well, I think that it's actually one of the most fascinating natural populations out there to study, because you can look at not just evolution, but also ecology side by side. You have hundreds of species interacting simultaneously. And just from a fecal sample, you can grab all those species DNA. And because of the timescales on which evolution and ecology are happening, they're actually happening side by side. And you have the opportunity to see eco-evo type of feedbacks. But also, you can measure evolution happening in multiple hosts simultaneously. And that basically makes us all in this room, all 50 of us, walking replicates of the same experiment. And that allows us to draw general principles of what's happening in this really complicated system. But finally, not limited to, it's also very motivating that the microbiome, and particularly the genetic variants in our microbiomes, have really important effects on our health and our phenotypes. Genes and SNPs in our microbes are responsible for modulating the way we digest food, conferring antibiotic resistance, changing the way we metabolize drugs, and even causing inflammation in our gut. Being able to understand the ecological and evolutionary processes that give rise to these variants in our guts will be important for precision medicine as we go forward. So, how do we study evolution in the microbiome? Well, let's start thinking about what we know about evolution from the test tube. Microbes have been studied for decades, and we have lots of insights about how they evolve in the lab. For example, Lenski and colleagues showed that E. coli grown in a test tube can evolve repeatedly and rapidly on short timescales. But this is a community composed of one species. What happens when you go to a community composed of hundreds of species? Are you going to see the same rapid evolution? Instead, let's say this person has an antibiotic. Instead of responding to that antibiotic via an adaptation, you might see it respond via a species fluctuation. Here you see the frequencies of E. coli and B. vulgaris shifting, and this has been quantified numerous times in the literature. So sure, ecology really matters in the microbiome. Another way the microbiome could respond to that antibiotic is via a strain invasion. I may not have an E. coli with an antibiotic resistance gene, but my neighbor might, and so I might acquire it from my neighbor. That's not necessarily evolution. What I'm defining evolution to be is a genetic change conferred by a mutation or recombination event on a pre-existing haplotype lineage in our guts. But finally, we might actually see the gut respond to that pressure via the administration or via evolution. So you might actually see a de novo mutation arise and then go to high frequency. And given estimates of billions of mutations coming into our guts daily, this is not an unlikely scenario. But as of just, I'd say 5 or 6 years ago, it wasn't really known whether evolution is common in the gut microbiome, or even happens for that matter, especially in the absence of a selective pressure. And in fact, to date, to 5 years ago, and even today, a lot of the quantification of our microbes are at the species level and more increasingly at the strain level. But so the question is, that we asked back when I started working on this topic in 2016 was, does evolution happen in the gut microbiome? And the answer is yes. Since then, myself with my colleagues, Benjamin Good, Oscar Halachik, and Katie Pollard, we published one of the first papers showing that evolution can happen in the human gut microbiome in healthy hosts or in any host. And this was really exciting. This was happening on just a few months timescales. Later on, I went on to show with my undergraduate that this can happen on daily timescales. And so this is really exciting because it means that there's a lot of dynamism in our guts, not only at the species level, but also at the genetic level. And this has a lot of potential to shape our health. Other papers have come out showing the same with different technologies. We use short read technology, but people have used long reads, high C reads, read cloud technology, isolate sequencing. But I argue that given the ubiquity and the in the affordability of short read data, if we can develop statistical methods to leverage that kind of data, we can unlock even more mysteries about how evolution proceeds in the microbiome. So given that now that it's kind of fact that evolution does happen in the gut microbiome, we can start to understand that from different angles. And so my group has been looking at questions like, how do within host microbiome evolutionary dynamics change over our lifetime? And how do microbes evolve across hosts? We can think about what's going on inside of us and in this whole community of us. So that brings me to my overview, which is as follows. So first, I'm going to be talking about the volatile ecological and evolutionary dynamics in infancy and how that gives way to strain stability in the adult gut microbiome. And then we have strain loss over our lifetimes. And then microbiome evolution across hosts. So we're going to look at this on a time scale. But it's not just about what's happening inside of us over a lifetime, what's happening across us. It's also trying to uncover the underlying evolutionary and ecological principles that guide us to conclusions on how our microbiomes are changing over our lifetime. So we're going to talk about how there's rapid evolution mediated by de novo mutations and recombination, and how there's an ecological model of stability that we can leverage to kind of quantify what we mean by stable. And talk about the community, that community diversity begets strain diversity. We'll talk about how there are limits to within host adaptation and stability. We'll talk about how horizontal gene transfer can facilitate adaptation across hosts. And then we'll also finally touch on the demography of our gut microbes and the deleterious mutations of their, the deleterious effects of their mutations. All right, so strap in. And we're going to start with the infant story. What's happening when we're just born? So how does evolution in infant microbiomes differ from that of adult microbiomes? This work was done by first author Daisy Chen. She was an awesome undergrad in my group, now a PhD student at UC San Diego. And when we started this project, we hypothesized that the microbes in babies might be evolving differently because they have different diets, different immune systems, and even different microbiome community members. My group uses a lot of publicly available shotgun data. There's just so much out there and so little understood about it. And so we looked at 4 data sets comprising 700 infant shotgun metagenomes spanning the first year of life, as well as moms sampled at time of delivery and healthy adults. And because these are shotgun metagenomes, we used techniques that I previously developed in my paper to look for evolution in these samples. So let me tell you a little bit about shotgun data. How do we detect evolution? So 16S data is really popular and really affordable for measuring species diversity. And it basically barcodes which species are present. But what it misses is all the different genetic information in the rest of the genome. And so you can't really see evolution with this kind of data. Instead, we can take a poop sample, isolate the DNA, shear it up, and then sequence that. And that gives you a kind of a snapshot of all the genes and variants in the microbiome. But this data is really tricky because these reads are really short. They're only 100 base pairs long. And we're dealing with hundreds of species. So it's like as if I took all of your DNA, mashed it up, and handed it back to you on a hard drive and asked you, tell me whose DNA is who and who is evolving what. That's really complicated. And so the task at hand is to look for these mutations occurring in one species at a time while dealing with the complexity of the data. So we need to actually simplify the data. And what we do is we bin reads by species. So in other words, we're mapping these reads to reference genomes. And then we can actually look in each species one by one. And here, for example, you'll see a mutation at 50% and be able to look at which mutations are present and at, their frequencies. In this way, we can then look across hosts and look at the same species, let's say. And you can see at this site, we have 80%, 30%, etc., and start to understand the variation, not just within a host, but also across hosts. And this kind of data has been really useful for tracking strain transmissions, for example, for mom to baby, whereby you look for alleles that are private to mom and baby and absent from the broader population. I don't have time to delve into this paper, but my PhD student who recently graduated, Leah Briscoe, developed an approach to perform source tracking with SNPs in the microbiome. And she was able to show that infants in the NICU have microbiomes that come from all sorts of parts of the NICU, including the floor, touch surfaces, the sink, etc. And so these SNPs are really useful for telling us fundamentally, where did our microbiomes come from? But going back to the question at hand, how do we detect evolutionary events? This is not so simple. Ideally, what we want to do is look for mutations going from low to high frequency over time. But can you think of another process that can actually generate the same kind of frequency change? Strain fluctuations. So you may not have just one strain of E. coli. You might have 2 or 3, and they might be genetically distinct. So we have a blue strain and a red strain, and they might fluctuate in frequency over time. That's not evolution as per what we're trying to find here. That's just ecological shifts in different players that can coexist side by side. So what we want to do is resolve that lineage structure to kind of delve a little bit more into what the lineage structure looks like inside of us, because we're going to return to this. Different hosts have some handful of strains per species. So I might have 2 strains of E. coli, and you might have 3 strains of E. coli. And interestingly, these strains are typically distinct across hosts. So my strains are going to be different from yours, and they've been actually called a microbial fingerprint. So this is a really weird and interesting distribution that we're going to revisit later in the talk. But for now, what we're going to do is resolve the population structure to be able to distinguish evolutionary events from strain fluctuations. But for the purposes of time, I will not go into how exactly we do that, other than we pick samples with simple population structures, and then we're able to track evolution confidently. All right, so we're going to now look at data. We're going to see, how does evolution change as a function of your first year of life? On the x-axis, I'm showing the transition of bacteria from moms to babies. Then I'm going to show what's happening in the first week of life, the first month, the first year. And I'm going to contrast that with what's happening in adults sampled on 6-month timescales. And on the y-axis is the rate of SNP change per day per base pair. And this is what the data looks like. This is so surprising to us that we would see, if you convert these to numbers and log it, you actually get a line. And so what it suggests is actually early in life, our bacteria are really rapidly evolving, and there's just a lot of volatility. And later on, this volatility gives way to a lot more calm, but there's still stuff happening. Do bacteria need to rapidly evolve to survive in their new environment? Possibly, given the diet differences, immune differences, the community membership that's changing, and this may be a way for them to survive. We dug deeper into what's happening inside of an infant to try to understand, well, how are these evolutionary changes happening? What is the underlying evolutionary mechanism? Are these seeded by de novo mutations, in which a spontaneous mutation arises, or by pre-existing variation that's already hanging around and maybe gets passed around by recombination? Or maybe mom was a reservoir of pre-adapted lineages that then got transmitted to the baby. So we explored all 3. And the way we're going to look at this is by looking at the prevalences of allele sweeping in infants. And the prevalence is as a function of the adult population. So here's what the data looks like. In gray, we have an expectation assuming only de novo mutations. And so basically they're all rare in the population, like no adult has them. They're private to the baby. But what we see in the data is actually a surplus of mutations that are completely ubiquitous in the adult population. So I think what we're seeing is a mixture of de novo mutations and some bad mutations that got into the baby and then reverted to a state that's favorable in the broader population. And we do see anecdotes of that where you'll see reversions over time. Moreover, you'll see if we look at the DNDS, which is the ratio of non-synonymous to synonymous mutations for these different categories, here we have a value of around 1 supporting the de novo hypothesis, and then a value of around 0.4 supporting the fact that these are common sites that are changing. And we can contrast this with what's going on in adults. You see a very different distribution. You see a whole bunch of mutations that are quite common. And they also have a DNDS of around 0.4, indicating they're not new necessarily. And so what we think is happening is that they're actually recombination seeded sweeps. They've been hanging around, experiencing purifying selection, and then sweeping. And indeed, when we look at the infant microbiome, we actually see a tremendous amount of recombination facilitating horizontal gene transfer. We see gene gains, we see gene losses, and this is all happening as the microbiome matures. And so we think that there's actually a whole range of evolutionary dynamics happening in the infant gut microbiome that can explain what's going on. And it's not just infants, it's also adults. And then finally, we took a look at strain replacements. You might have a strain of E. coli, and your family might give you a different one. And what we see is, again, just when the baby is born, a tremendous amount of turnover and volatility in terms of strains. Maybe some are not fit enough, they get replaced, or maybe it's stochastic. But you can see this gives way to stability in adults, and that's a good thing. You don't want your microbes to turn over every day. If they just finally figured out how to digest that burrito, you want to keep them. So we're going to now delve into this to understand how the volatility gives way to stability. So we're going to talk about quantifying the stability and, moreover, how we get these strains, this particular distribution of strains in our gut. And so we're going to ask 2 questions. Are the strains stable? And why this distribution of strains? So we'll start off with the first question. And this work was done by Ricky Wolf, who's right here, a really awesome PhD student, and a former postdoc, William Shoemaker. And we looked at the ecological stability of strains using 4 dense time series coming from healthy adults that were sampled basically every day. And if you take a look at 2 of these host microbiomes at 2 species, on the x-axis is time, on the y-axis is the SNP frequency, and Ricky spent a tremendous amount of time phasing these strains and tracking their trajectories. You can see that actually there's a lot of, well, firstly, this guy has 3 strains of B. vulgatus. This guy seems to have 2 strains of F. prausnitzii. You can see that they kind of jiggle around this carrying capacity. But in this case, they don't. There's like this wild swing. They flip. So let me ask you, which one would you say is stable just from your naked eye? B. vulgatus? Raise your hand. Or F. prausnitzii? All right, we're on, our eyeballs are doing our job for us. So that's good. So, but you can see there's bouncing, right? There's fluctuation. Is that fluctuation what we expect under stability? So we can try to quantify those fluctuations using a stochastic logistic model, which describes the abundance fluctuations around a fixed carrying capacity. And we can ask, does this model describe the data? And so how does this work? You have some growth to this carrying capacity shown in the dashed line here. Then you can have some noise driven by the environment and also randomness. And then if you simulate those thousands of times, you'll get basically everything settling out to the same carrying capacity, more or less, with some jiggle. And so if our data falls into this red part that's stable, then we can say that yes, our data is consistent with a model of stability. And so going back to the data, Ricky fit the data to the first third of the data, model, so mean and variance, and asked, can that describe the entire trajectory? And in the case of Vivo Goddess, it can. But in the case of F. prausnitzii, it cannot. And so that was really reassuring. Our eyeballs are doing the right job for us, but we can also quantify this with a model and reject or accept stability. And so if we look at this across all 4 hosts we analyzed and all their species, we can say that in the majority of cases, roughly 80% of hosts by species combinations, the SLM is sufficient to describe dynamics. So what does that mean? Adult microbiomes are stable. Hooray! You don't want to lose your microbes that easily. Okay, but we're going to return to this point of stability in 2 minutes, so keep that in mind. And we're just going to switch gears for a second and now talk about this really funny distribution of strains inside of us. As I've been saying, we have this weird distribution of 2 strains of E. coli in me and 3 in you. Why this distribution? Why isn't it exactly 1 or hundreds? This question actually continues to keep me up at night, and I think is a question we're going to have to keep chipping at as a field, but we can start to try to understand it and try to understand what are the kind of correlates with the strain count. So this is a collaboration with Professor Jesse Shapiro at McGill University, his student Naima Mahdi, and then Daisy and Ricky, who I've introduced. And we asked, is community diversity correlated with intraspecies strain count? So let me introduce a few hypotheses that we explored. So on the x-axis, we have community diversity. So in other words, how many species are present, or what's their alpha diversity? It could be low, or it could be high. And if it's low, maybe there's not much competition in the microbiome. And so without that competition, you don't have metabolites being produced and new niches being opened up. It's kind of like a little boring. But in the high diversity one, you have a lot of competition, interaction, cross-feeding, excess metabolites being produced. And so what could happen then is you open up the opportunity for new players to come in. And so in this scenario, you have a strain invasion for the high community diversity microbiomes. So that's called the diversity begets diversity hypothesis. In contrast to that, we have the ecological controls hypothesis. And so here you have only a few players. And so actually you have a lot of niches open. So that invites more players to come in. And here all those niches are filled. So which one are we going to see? Are we going to see this positive correlation with strain count and community diversity, or a negative correlation? Well, let's take a look at the data and see what we get. So same X and Y axes, and we took a look at this many different dimensions, and what we got is a positive relationship supporting the diversity begets diversity hypothesis. So what that means is that the more diverse your microbes are, the more intraspecies variation you're going to have. Interestingly, we also saw some dynamics with gene loss. as your community diversity gets larger, you also see an increase in the loss of genes. So in other words, let's say you live in a dense community and your neighbor starts making cookies. Why should you continue to have that cookie gene when you can just eat your neighbor's cookies? That's called the Black Queen hypothesis, where you take advantage of the community goods and you shed your genes to become more efficient. So it seems like the community diversity is really playing a role in shaping interspecies diversity. Now, I'll try to get to this a little bit later as well, but this Black Queen effect could have some really weird consequences later on. In a collaboration with Will Ludington, we're exploring the hypothesis that the bacteria shed genes, but then they, let's say that cookie maker moves away, then you don't have any cookies, and then you're going to die. And then whatever you were making, maybe as brownies, your neighbor's going to die, right? So then the whole community starts to destabilize. And what happens then is that you then have pathogens coming in and maybe causing inflammation that in old age maybe causes all sorts of diseases. And so this is something we're testing in a model of aging, TBD. So as I said, we have volatility in infancy that gives way to stability, but then that gives way to strain loss. So what we want to do is not just look at our microbiomes on daily or monthly timescales. We actually want to look at this on decadal timescales. So the ideal data to test long-term dynamics is to sample the same host over a few decades. But a PhD lasts much shorter than a decade, fortunately. So what are we going to do instead? We're going to leverage adult twins to test for long-term dynamics. So previous literature, and actually many papers now are showing that cohabiting individuals, not just siblings, share many strains. So we found this data set of twins that grew up in the same household, moved away to college, lived apart for 40 years, and then were sequenced. And we asked the question, do adult twins share the same strains or not? If they do, it means that what they grew up with is what they have in adulthood. And if they don't, it means that what they grew up with is lost. And the answer was no. Basically, there was virtually no sharing between these twins. And that was really surprising and exciting because it means stability is ephemeral. Eventually, all strains are replaced over 20-year timescales. It also means that there are limitations to adaptation within a host. The bacteria cannot continually evolve to adapt to our host genetics and then stick around forever and then get passed down to their future generations of kids and so on. They're going to be replaced and pulled back into the broader circulation of strains, and you're going to get new ones. Why are strains replaced? Well, maybe this destabilization of microbes due to Black Queen effects. We've looked at antibiotics. It doesn't seem like bacteria are wiped out. They actually rebound. And so that's also an avenue for future research. How does strain loss happen? And why? All right, so now we're going to look at microbiome evolution across hosts. We've been looking at it within hosts over timescales of days, weeks, months, and years. But now we're going to look beyond our lifetimes. We're going to look at what's going on timescales maybe before, that happened long before we were born, and quantify evolution from that lens. So we've been looking at adaptations that can show up within a host. Could it be that the same evolutionary change arises in multiple hosts? We all go to lunch together and we have the same burger and we all evolve the same adaptation to digest that burger. Could it be that we're individually doing that work again and again? Well, let's take a look at what that might look like. So we have here many hosts, and each horizontal line is a haplotype corresponding to their bacterial genome. We have a beneficial mutation arising in one host, and then it shows up in 3 more hosts. You can see that the genomic signatures don't really change around this site. And so the question is, do we see this evidence of recurrent de novo mutation in the data? We see some limited empirical evidence. In infants, we saw some recurrent mutations in the same pathways, but not really, actually. And maybe that's having to do with their detection limits. We need to develop better methods to see such mutations. But instead, we actually see a lot of purifying selection happening between hosts. In fact, the stuff that's arising within us seems to get weeded out over long timescales. If you look at this through the lens of DNDS, or the ratio of non-synonymous to synonymous mutations, if you look at my strains and your strains, you'll see that in each dot here, we're looking at this at a genome-wide level, at a pathway level, all sorts of levels, and we always see this statistic which can quantify evidence of adaptation settling out to a low value of 0.1, which kind of indicates actually a lot of purifying selection. So it doesn't seem like there's a lot of evidence or much for recurrent de novo mutations yet. That's TBD. So is there evidence for a cross-host adaptation? Well, maybe it's happening via a different mechanism, and it's not recurrent de novo mutations, but rather there's migration. My bacteria has evolved the mutation to digest the burger, and it then migrates to all of you. So then what kind of genomic signature should we expect from that? Ideally, in this kind of scenario, we'd see the same genomic backbone in everybody. Everybody would have the same exact strain of B. vulgaris or E. coli. Does that happen? No. Remember, everybody harbors their own unique strain. You have your own microbial fingerprint, right? Your microbes are special. So this cannot be the model of what's happening. What else could generate the spread of an adaptive mutation across hosts? Migration and recombination. Remember, we saw a lot of recombination happening inside of hosts. Genes are coming in and getting lost. Could it be that this recombination is facilitating the change, the spread of this adaptation across hosts? We think so. And here's Ricky again. He's been working really hard on this paper, so stay tuned. It's going to come out really soon. And if you want to read it in advance, we'd love your feedback. And we're really excited actually about this paper. And so what we think is happening is that there's gene-specific sweeps going on. You don't see dips in diversity genome-wide, but you see it in the vicinity of the adaptive mutation. And importantly, the linked region, neutral and deleterious variants, also go to high frequency. So it leaves these characteristic signatures of haplotype homozygosity right in the vicinity of the sweep. I won't go into the details of the statistic that we've developed, but we're very excited about it. It essentially leverages linkage between non-synonymous sites versus synonymous sites, capturing this tight correlation between these red and green dots. I'm happy to take more questions about it. And when we apply this statistic to microbiome data, we get some really interesting candidates of selection. So this is the species Ruminococcus brahmi. In orange are significant hits. You can see these peaks. And Ricky's highlighted this MDXEF locus. What is it? Well, it's involved in maltodextrin metabolism. What's that? Well, if you've had potato chips today or like at this seminar, you probably had it. It's an additive, a starch additive that comes from these potatoes. And so it's actually something that's been added to our food. And we probably saw a lot of change in our diets due to this and other changes due to preservatives over the past 100-ish years. So our diets have changed, and probably our microbiomes have also changed. Isn't that cool? It's kind of like the lactase persistence story, but in your second genome. So we're really excited about this. And Ricky took a look at the haplotypes. So these are different hosts. And you can look at the signatures right around the locus under selection that we think is under selection. And you can see there's a lot of homozygosity, and it really resembles a gene-specific sweep, like what we hypothesized to be happening. So stay tuned for that paper. But not all mutations are beneficial. Remember, there's a lot of purifying selection, and in fact, that you can even have some of these bad mutations going to high frequency. And so we talked about how a lot of mutations are coming in, getting weeded out. And so this brings me to a project in collaboration with the really awesome John Ma in my group right here, and Professor Kirk Lomuller. It's been a joy to work with on. And we looked at the inference of distribution of deleterious fitness effects. So how do we infer a distribution of deleterious fitness effects? Or in other words, what is it? Well, we're trying to measure what is the typical deleteriousness of a mutation. Is this going to be lethal? Is this going to be tolerable? How do we do it? Well, we, in a nutshell, take a summarizing statistic of the data called a site frequency spectrum. It's a distribution of allele frequencies across hosts. And we're looking at this across the whole population of hosts, not within hosts. We need to actually infer a demographic history of these microbes, which is an added really awesome bonus, and truthfully was what drove this project to begin with. And then we can finally infer this distribution. How many mutations are really bad and how many are benign? So let's take a look at the demographic history that we uncovered in this process. So here on the x-axis, I show how the population size is either contracting or expanding over time. And on the y-axis, I show years ago. If you notice, these are all significant epochs in human history. 10,000 years ago was when agriculture started. 1,000, roughly, things are starting to change in human industrialization. 100,000 you see out of Africa. And this is where what we see in the data. We see demographic changes spanning the entire course of human history. And hey, guess what? Look at that species, Ruminococcus brahmi, who is involved in degradation of starches, shows an expansion a little bit roughly around the time of agriculture. So maybe our microbes experienced changes in their population sizes corresponding to diet. Well, this is a TBD and future food for future work. Okay, so now let's look at the DFE finally. What's a typical effect of new mutations in the microbiome? So we can take a look at this species, Bacteroides theta iota micron, and we see a shape that looks like this. So a lot of really deleterious mutations. We can look at this across the 25-ish most common species, and you can see there's a lot of variation. But if you squint within clades, you'll see some similarities. Like this Eubacterium clade has a bunch of really deleterious mutations, whereas these Bacteroides species have mutations that are less deleterious. Why? This is also an avenue for future research, and maybe this means that there are different versatilities of bacteria able to tolerate different environments across hosts. In fact, we've been basically looking at this DFE using kind of an average across hosts. It could be that DFEs are varying from host to host. What is beneficial to me might be really deleterious for you. And remember, in infants, we saw those reversions. maybe what's beneficial in mom is bad in baby. So there could be fitness trade-offs that would be very interesting to explore. Okay, so I want to conclude and then share a little bit more about the broader vision. So today, what we talked about is the evolutionary and ecological dynamics of our gut microbes throughout the course of our life and beyond our life. We looked at the volatile ecological and evolutionary dynamics in infancy, how that volatility gives way to stability in adults, how there's limits to that stability, and we see strain loss over our lifetimes. And then we also looked beyond our lifetimes at evolution across hosts. In the process, we uncovered a lot of basic ecological and evolutionary principles. We saw that evolution inside of a host can be mediated by de novo mutations and recombination, and also pre-existing variation that's circulating in mom, for example. We looked at an ecological model of stability, and we saw that community diversity begets strain diversity. We saw that there are limits to within host adaptation and stability, that there's adaptive horizontal gene transfer, and we uncovered the demography and the DFEs of new mutations. And this work was really pioneered by the people in my group, and some of these points are actually brand new in the literature. Nobody has ever uncovered the demography of these species before. Or this is, the very first scan for selection across host microbiomes that systematically identifies targets of selection. And so we're really excited actually to share this work with you today. Now, going more broadly into what is the research question in general, how do we understand adaptation in natural populations? A big question that really interests me is how rapid is evolution in natural populations? In infants, we saw evolution happening as short as on a daily time scale, which is wild. There's evolution happening right inside of you right now. But how does this rapid adaptation translate to non-microbial populations? And I'll take a moment to go back to where I began my career, which is looking at signatures of gradual versus rapid adaptation in Drosophila. We looked at signatures of hard sweeps associated with gradual rapid adaptation and soft sweeps associated with rapid. And I don't have time to go into this deeply today. But the point is, these are signatures from genomic data that can tell us about the rapidity of adaptation. And PhD student Mariana, who I saw, she's right there, really awesome PhD student and a joy to work with, has been exploring how different parts of our genome can have different rates of evolution. For example, autosomes are, I showed in my previous work that autosomes have a lot of soft sweeps. They're experiencing rapid adaptation. What about the X chromosome? Should the X chromosome produce harder sweeps than autosomes? And we think they should, because of lower effective population sizes, more efficient positive and purifying selection. And through a lot of hard work, Mariana showed in both data and simulation, not just in melanogaster, but in 6 Drosophila species, that there are harder sweeps on the X. And so, how common are hard and soft sweeps in the microbiome? Stay tuned. These are the dots we're connecting in our lab, and we'll be excited to share this with you as it unfolds. So that's just to kind of give you an overview of what we're doing. I'd like to acknowledge the many, many people who contributed directly to the work that I presented today. These are members of my group, but not the only members. I will share with you more. I'd also like to thank my collaborators, past, present, and I'd also like to thank my former advisors, Katie Pollard and Dmitry Petrov, and my funding for putting their faith behind us. And I'd like to thank my research group for being really a joy. They made coming to work every day really exciting. And they have just an enormous amount of creativity and drive and technical prowess. It just is really fun to watch them grow these ideas. I'd also like to thank Michael Wozni, Peter Lauren, Aina Martinez, Zurita, who I didn't mention today. They are more recent recruits in the lab and working very hard on some really exciting projects that we're really excited to share with you in the coming years. I'd also like to thank the EEB department for giving me the chance to start my lab here. It's been a lot of fun being amongst you. I'd like to especially thank Kirk and Jamie for their mentorship and reading all my grants. Allison Peter, who also was a very important sounding board for me. She read a lot of papers that first drafts. CompMed colleagues for helping me also with grants, and Ashwath Raman for his unwavering support and inspiration. And so with that, I'm happy to take questions, and thank you for letting me present.
I'm always good for a question. So I'm just curious, of course, transcending to larger ecological problems. And I thought maybe the first step on that would be to study people and their pets.
Yeah.
Do people do that?
Do people study people and their pet microbiomes? They do. And guess what? You probably look like your dog. No, but it's true. Like there is a kind of cross movement of.
And so you can expand from there potentially.
For sure. Yeah. Yeah. Yeah, for sure. Yeah, Victoria.
Wonderful talk. It's really great for me to see a synthesis of what you've been doing. So this is sort of a very simple question. How much does genetic drift? You see how different the bio is. I would think genetic drift would be playing a really great role. And how does that weigh in terms of differentiation among individuals versus deleterious versus adaptive responses?
Thank you for asking the question. To repeat it for Zoom people, how much role does genetic drift play as opposed to purifying selection or maybe adaptation? So the story I told about John's paper on this inference of DFE really did not do justice to the paper. And I also should add the paper's just about to come out in bioRxiv, maybe next week. So if you want to read a draft, it's there for you. So what did we answer in that paper? We actually find something really interesting. And I'm going to first address it from the across host perspective. We've been focusing on how deleterious are mutations. And what John did is he divided up the genome into core versus accessory. Accessory are genes that are present in some microbiomes, but not all. Core are basically genes in everybody. And he found, logically, that actually the effective population size for accessory is lower. Makes sense. Fewer copies of that gene. What does that mean then? It means that if your effective population size is lower, then the effective drift is stronger on these genes as opposed to purifying selection. So that's one way to answer your question. The other way is to think about the effective drift inside of us. And the kinds of evolutionary changes that we're looking at are large swings in allele frequencies from low to high over short periods of time. And it's unlikely that in such large populations inside of us, that drift would be the dominant process generating those swings. But for sure, there could be a lot of stochasticity happening, and it's just harder to see with shotgun data. And I think that is definitely an avenue for future research. Yes.
I was pretty interested in your point that community diversity begets strain diversity, and that seems to say that community diversity actually has a causal relationship with that. But I'm wondering if it's just the mechanisms that underlie propagation of strain diversity just operate on a broader time scale and are a little bit slower than I assume just the extinction and the colonization dynamics that govern the community diversity. Like, do you see that as being a causative relationship, or they're both just a product of stability?
That's a great question. So I'll try to repeat the question. Is community diversity causal of strain diversity? Or could they just be happening side by side on different timescales? and being a product of stability. I don't want to claim causality. And in fact, in this talk, I'll admit, I use the term, we saw diversity begets diversity. You're right to conclude that point. But in reality, in the paper, we toned it down a lot. And it's actually correlative, right? So whether it begets that is, I think, something that should be probed in the lab, and people are probing it in the lab, where you systematically change diversity and tune that knob. And so, as I said, how we get this interesting distribution of strains is something that we need to continue to probe. Maybe it's because the strains are kind of genetically complementary, or maybe it has to do with who you're encountering. It's a, or the foods you eat. So there's a lot there to study. Yeah.
I'm curious about the twins and like how you sort of have different microbiomes, but how different are we?
Yeah, so the question is, how different truly were the twin microbiomes? They were different strains. Like really, so the alternative hypothesis is that me and I don't have a twin, but let's say two twins, they get the same strain at birth, and those strains diversify continually, and then when they're 60, those strains look really different when they really had a common ancestor, recent ancestor. But if you compute the product of the mutation rate and the generation time and the longest time span that a person can live, you cannot get the number of mutational differences that we saw. So in other words, I think over a lifetime, you could probably get a few hundred mutational differences between, your strain at birth versus your strain at death. But if you look at the number of mutation differences between two strangers, you're going to see that on the order of 10,000. It's not feasible to generate. They'd have to like some kind of cancerous bacteria or something. So it's we were pretty sure they were different. So thank you. Yeah.
So this idea of strain loss over our lifetimes is really interesting. I'm wondering if you've noticed, or maybe you've seen it in other papers already explored, but if there's a difference in the rate of strain loss between men and women, and if there is, why that would be.
Have I seen a strain loss rate difference between men and women? I haven't, but it's an important question to ask. Are there sex-specific differences? And I know people in the field are researching that from different angles, whether it's the metabolites that the bacteria are producing and so on. I don't have an answer for you. It's worth actually investigating, maybe in the context of, different cohorts like preterm birth and so on. Yeah.
Just curious, looking at strain loss and the things you're looking at, do you see hygiene being a big impact of being able to kind of spread it to hosts?
Can you say it one more time, please?
Have you seen a major effect the development of hygiene? Most of us probably wash our hands with soap.
Oh, hygiene.
Probably bacterial. And so is that potentially driving some of the trends we see, especially strain loss, if we're buffering ourselves using this kind of external?
Yeah. So what role might hygiene be playing in strain loss? So there's something called the hygiene hypothesis, where people, especially in industrialized communities, are washing their hands, having antibiotics, and living in very relatively to speak more sterile conditions. And so John kind of explores some of this in his paper, and sees on long time scales from movement of bacteria from Africa to North America, rural Africa, you see a tremendous loss of bacterial diversity. And we're not the first people to see this. Many people have shown that this hygiene plays a role in loss. But that's over long time periods. What's happening inside of us, I'm not sure if there's a connection. For example, antibiotics is something that's a huge perturbation to our system. It's like a shock. and ideally should kill off the bacteria. But interestingly, the bacteria rebound. And I read this really fascinating paper that showed that the bacteria are adapting to burrow into the gut, but not to adapt to the antibiotic. And they kind of like seclude themselves while the antibiotics is playing its course, and then they come back. And they're still susceptible to antibiotic, but they have the ability to move around and hide. So on a lifetime, I don't know if that's the cause.